What, Vam? Two last wrinkles I wanted to talk about are density-dependent selection and a balanced polymorphism. I didn't have time to add slides illustrating a balanced polymorphism, but you actually saw one on Monday when I was messing with that population genetics simulation. Polymorphism just means, it's Greek for many shapes. And in genetics, in this context, it means many alleles. So a polymorphic locus is just a gene that has multiple alleles. And a balanced polymorphism is what you get when selection favors heterozygotes at the expense of both homozygotes. The example that I gave in class was sickle cell anemia, where if you are homozygous for the sickle cell gene, you have a disease. And without supportive medical care, you may not live a full life. However, if you are homozygous for normal hemoglobin, you're susceptible in some parts of the world, notably West Africa, to getting malaria. And if you are a heterozygote, you don't get the disease and you have some degree of protection from malaria. And malaria is one of the global killers. Up until recently, it was estimated to have killed perhaps 800,000 people each year in areas where malaria is endemic. So that's the reason why selection hasn't eradicated the sickle cell allele. It's because selection favoring heterozygotes keeps it in the population. And what you end up with is a stable frequency of the sickle cell allele in West African human populations. That's a balanced polymorphism. Density-dependent selection is what you get when the fitness of a genotype isn't constant. It's a function of how common that genotype is. What the heck is that? These are the best pictures I could get of a fish that is native to Lake Tanganyika in Africa called Parasodus eccentricus. This individual up at the top, you can see that the mouth is kind of open more to the fish's right side than the fish's left side. You can see more of the mouth over here than you can over there. This fish, you can see more of the mouth on the fish's left side than you can on the right. This is a fish whose mouth is twisted to one side or the other. And the reason is that this is a fish that is lepidophagous, meaning it feeds on the scales of other fish. There's more than one species of fish that actually does this, makes a living this way. We'd call this a micropredator. Now, fish have very wide fields of view, so they're not easy to sneak up on, as you may have noticed if you've ever had to noodle catfish. So this fish sneaks up behind, and its mouth is twisted either to the left or to the right, so that it can approach from behind and to the side and grab a mouthful of scales with a minimum of disturbance. So we have some Parasodus that have their mouth twisted towards their right, and some Parasodus that have their mouths twisted toward their left. And it turns out that the frequencies of left-handed and right-handed Parasodus fluctuates from season to season. You can see that it zigzags fairly evenly. And the reason is this. Let's say we have a bunch of Parasodus that had their mouths twisted to attack from the left. Well, the prey is going to start getting sensitized to threats coming in from the left. Fish are going to start being sensitive to and avoiding anything sneaking up on them from the left, right? I mean, correct? So if the lefties are very common, fish become learn to be wary of them. That gives the righties an opportunity because their prey isn't watching out for them. So when the prey fish start getting wary of attacks coming from the left, that leaves them vulnerable to the attack of fish coming in from the right because that's not what they're expecting. So the righties are favored. But then as righties become more common, their prey learns to avoid the righties. And now the lefties that are more rare have the advantage because that's not what their prey is expecting. So what selection is doing is favoring whichever phenotype is more rare. Of course, if selection favors a rare phenotype, that means the rare phenotype becomes common. And once the rare phenotype becomes common, selection no longer favors it. So the point here is that selection favors whichever phenotype is least common. And because selection will take that least common phenotype and make it more common, then selection tends to alternate favoring lefties versus righties on a cycle that looks like it lasts roughly 5 years. You can see we have these fluctuations between lefties above and righties below that looks like it goes through a cycle roughly about 5 years long. So that's density dependent selection, where the fitness of a genotype doesn't only depend on, whether the fitness of a genotype depends on how common that genotype is. There's another example. It turns out that Drosophila, fruit fly larvae, have two types of behavior. They can be rovers or setters. The rovers will tend to move long distances when they feed, and setters tend to stay in one place. And this happens to be governed by one gene with two alleles. Density-dependent selection maintains both of these in the population. If setters are common, that means the setters are staying in one place as they feed, and they're likely to exhaust the local food supply, and the rovers have an advantage because the rovers are leaving and finding new sources of food. So when the setters are common, the rovers are favored, and they'll breed and they'll lay lots more rover babies. And then when the rovers are common, the setters are favored because the setters stay in one place and don't spend a lot of energy roving around trying to find new sources of food and possibly failing at it. So when rovers are common, setters are favored, and when setters are common, rovers are favored. Selection favors whichever allele is rarer in the population. That's density-dependent selection. Okay, last thing I want to cover for today is selection operating on continuous traits. And this was something we briefly mentioned when I was talking about the rediscovery of Mendel's laws. And I'm going to start with something that is controversial and possibly potentially personally a bit upsetting. This is certainly not something I agree with, but I would be a fool to pretend that it doesn't exist. This was a book that came out in 1994 by a political scientist and a psychologist at Harvard. The political scientist at a conservative think tank called the American Enterprise Institute. And they published a book called The Bell Curve: Intelligence and Class Structure in American Life. Now, real briefly, what is a bell curve? When you have a trait that tends to be determined by lots of factors, lots of little things, the distribution of the trait tends to approximate a particular type of curve. If I was to measure the height of everybody at UCA, we'd have a small number of people who are really short, and we'd have a small number of people that are really tall, maybe our basketball centers or people like that. And most of us are kind of somewhere in the middle. And if we did like a bar graph with the number of really short people and then the number of kind of short people and the number of sort of short people, and the number of medium short people, and the number of average people, and the number of medium tall, and kind of tall, and really getting tall, and damn, how's the weather up there tall, we'd end up with a bar graph like this that would approximate the shape of a bell curve. We'd get the same thing if we were to measure, uh, I don't know, any kind of trait like this that depends on lots of different factors. If we went out and measured the intelligence of the population, gave everybody IQ tests, we'd find a few geniuses over here and a few dimwits over here, and most people are kind of somewhere in between, more or less in the middle. If we were to measure people's tendency to commit crime, we'd have a small number of maniacs over here and a small number of nuns over here, and most of us are kind of sort of somewhere in the middle. So we get that bell curve an awful lot, and you get it whenever you've got a trait or a feature that is determined by a large number of factors. For that matter, it doesn't have to be a genetic feature. Let's say we had 6 dice and we rolled them and we added up the sums. Very rarely all 6 dice would show 6 and we'd get a number 36. That would be very rare. We'd get 36 a small number of times. Very rarely all 6 dice would show a 1 and we'd get the number 6 right here a small number of times. Most of the time we'd get a number that was kind of in between 6 and 36. Most of the time we might get, I don't know, 17 or something like that, and that would be here in the middle. And the distribution of numbers that we threw would approximate a bell curve. Okay, what's this got to do with anything? Herrnstein and Murray argued, well, first of all, that intelligence as measured by IQ, if you measure a bunch of people and plot them out, you get a bell curve. There's a few geniuses and a few dopes, and most people are sort of in between. What they argued was that bell curve looks different for different racial and ethnic groups. Boom. There's a quote: ethnic differences in cognitive ability are neither surprising nor in doubt. Large human populations differ in many ways, both cultural and biological. It is not surprising that they might differ at least slightly in their cognitive characteristics. Intelligence. One message of this chapter is that such differences are real and have consequences. So there is one of their graphs. The NLSY is, I think that stands for the New Longitudinal Survey of Youth. It was a big long-term psychological survey of a large population. And among other things, they measured everybody's IQ. And in the population that they measured, when they measured the IQ of white people, the bell curve looked like this. We've got a small number of geniuses over here whose IQ is close to 150. We've got a number of people where it's down at 70. I think 70 is the cutoff for significant developmental delay. Below 70, I think you're going to have trouble taking care of yourself. And most people are kind of here in the middle. That's the bell curve for the white population in the NLSY. The black population also met a bell curve, which I'll trace. It looks like this. And notice that there are a few very intelligent black people in here, and there are some black folks of low intelligence. But the average right here, if it's a bell curve, the average is going to be equal to the mode, the most common value. The average is about 85. People of European descent, it's about 100, maybe a little above. So the IQ distribution of African Americans is about one standard deviation less than that of European Americans. I think you can see that this pissed people off. That stirred up just a little bit of controversy in some circles. In fact, this is a book that came out a couple of years later, rebutting the arguments in um, the bell curve wars, including an essay by evolutionary biologist Stephen Jay Gould. I might make you guys read it. Yeah, why not? We're not going to shy away from uncomfortable topics. And as you can tell, it bothered quite a lot of people. Um, you're dealing in very dangerous ground when you start asserting that certain ethnic groups are less X than others, that some ethnic groups are less intelligent or less gifted or whatever it is. Um, that gets to be a road down which you don't particularly want to go. Germany tried it in the 1930s and it did not end well. I certainly don't agree with it. I think the data is basically right, but the interpretation that the difference between white and black populations in IQ is real and genetic is utterly false. And I think I can demonstrate this, but it might be Friday before I can deliver what I hope is going to be a knockout blow. So let's delve deeper into this argument and see if we can't reevaluate this a bit. Um, Eventually, I'm going to come back to the bell curve. But right now, I want to talk just a little bit about, in general, how we handle evolution and heredity of traits that are continuous instead of discrete. We call these quantitative traits. They don't have a small number of phenotypes, they vary across a range. Um, Pea plants can be either tall or dwarf. Humans are not either tall or dwarf. We're not all either thoroughbred jockeys or NBA centers. Um, we have some people that are very short and some people that are very tall, and most of us are kind of in between. That's more or less a bell curve distribution. And so height, weight, length, size of body parts. If I was to measure the size of your head or the length of your arm or something like that, that would be another quantitative trait that varies continuously over a range. And intelligence, however you measure it, also varies continuously. You know, we're not all either geniuses or dummies. Most people are kind of in between, and then we have outliers in either direction. You might remember I mentioned there was a battle between biometricians and Mendelians in the early 20th century. Mendelians argued that you could reduce all of heredity to Mendel's laws. Biometricians pointing out that there were a lot of traits that didn't look like they followed Mendel at all. And the reason ultimately is that these quantitative traits are determined in part by the environment and in part by interactions among many genes, lots of them. One example, like athletic ability. There are a few people that are absolute potatoes and a few people that are Olympic class athletes, and most of us are kind of somewhere in between. Turns out, I think they've identified at least 200 genes that are directly linked with your athletic ability. There's no single gene that makes you either, you know, Simone Biles or Jabba the Hutt. What there is a lot of genes, and you know, at least 200 that are directly involved in aspects of how your muscles function. I wouldn't be surprised if the number of genes affecting intelligence is even greater. Skin color. There's at least, last I looked, I want to say there were at least 9 genes that affected your basic skin color, which is why that also varies continuously. We're not all ebony or ivory. We range from peaches and cream, all the way to black coffee and everything in between. Um, that's another trait where we're not all black or white, the way that pea flowers are either purple or white. We vary on a continuous range because we have a lot of genes that all have an impact on the tone that our skin eventually takes. And you can select on quantitative traits, and that tends to happen in 3 different ways. These you need to know. These are actually on that practice exam. There's directional selection, stabilizing selection, and disruptive selection. Directional selection moves the mean phenotype. Stabilizing favors the mean and acts against the extremes. Disruptive selection favors the extremes and acts against the mean. Let me show you what these look like. And let's go to the whiteboard real quick and clear it. Okay, let's draw okay, there's a graph, and on the y-axis, or on the x-axis, we have SOME. We have some measurement. It doesn't even matter what. And on the y-axis, we have frequency of that measurement. Okay, we've got some kind of trait, whatever it might be, and we take our population and we measure that trait in every member of the population, and we find that a few members of the population have a very small measurement, and most members of the population are kind of in the middle. And then a few members of the population have a very high value for that measurement, and we get a bell curve. Okay, doesn't even matter what we're measuring. It could be anything. If we have directional selection, here's what we do. We take, let's say, all of the individuals with a low value of the measurement, everybody that has maybe small height or small weight or small whatever it is, and we stop them from breeding, and we take all the big guys and encourage them to breed, and after a certain length of time, I'll use blue to represent what happens later, we might come back to the population and we'd get something like this. Okay, we'd still have a bell curve, but the bell curve has essentially slid in a direction, and the mean value has moved in a direction. We've taken that bell curve and we've shoved it. Okay, that would be directional selection. Selection that pushes the mean in one direction or another. We could push it in the opposite direction too if we wanted. That's directional selection. Now, let's see. Stabilizing selection. Let's say we have individuals with this trait, and all of the individuals that have a very small value for that trait, we don't let them breed. And all of the individuals with a large value for that trait, we also don't let breed. What we'd see over time is that bell curve getting more narrow. If we favor the mean and select against the extremes, then we decrease the amount of variation in the population, and that bell curve becomes narrower. That is, you can see the mean doesn't change, but the distribution does, the width of the bell curve does. That would be stabilizing selection, where you're selecting favoring the mean and disfavoring both of the extremes. And then the third possibility is the reverse of that. What happens if we encourage both the really small and the really large to breed and we don't favor the mean? What we would get over time would be something more like this. We'd end up with a curve that was a lot more wide. And this would be disruptive selection. Again, the mean doesn't change. Our average trait value, our average measurement is still going to be the same, but this bell curve gets a lot wider. It may even develop this two hump shape if all of the mean individuals are selected against and only the extremes are favored. That's disruptive selection. So there's our difference. And let's go back to the slides. There we have it. Directional selection moves the mean phenotype. Stabilizing selection favors the mean and acts against the extremes. Disruptive favors extremes and acts against the mean. And you could get combinations of these, be it said. But let's look at some examples. There's Geospiza fortis. That's one of the Galapagos finches. A couple of researchers at Princeton named Peter and Rosemary Grant have been going to the Galapagos every year for coming up on 50 years, studying populations of Galapagos finches. The Galapagos finches are some of the best studied populations in the world. And here's what they did. G. fortis is one of the large-beaked finch species. And they trapped a bunch of birds on one of the islands. I think it's the one called Daphne Major. They measured beak size. And in fact, they measured several beak dimensions and combined them into a single number called the first principal component. Don't worry about what that is. It's a way of combining several different measurements of size and shape into a single number that's easier to keep track of. Don't worry about how it's done. You can just think of it as a measure of overall beak size. And yes, we have negative numbers here. That doesn't mean that the beak didn't exist. That's just the way first principal components work. So don't worry about the fact that this is negative. We just need to know that this is a measurement of beak size. And you can see beak size stayed pretty constant in 1975 and 1976. 77, they got hit with a big drought. The Galapagos get hammered every time we have an El Nino year. Tends to correlate with a heavy drought in the Galapagos Islands. How does that affect the birds? Well, these are seed-eating birds. The plants that make small seeds tended to produce a lot fewer seeds. There was much less food. Birds with bigger beaks could crack tougher seeds that were likelier to survive the drought. So there was death. Birds died because they couldn't get enough food when this drought hit the Galapagos. And what that caused was a measurable change in beak size. You can see the curves for both male beaks and female beaks went up quite a bit during the drought. These dots right here are means. Those are average beak sizes. And you can see how the mean increased from this value to this value to that value during the drought. That would be directional selection. The mean beak size shifts. This would be stabilizing selection. This is kind of a complicated graph. So this is birth weight. The stair step thing right there, that's a distribution of birth weight in the population. Most people are born at roughly 7 pounds, right? As much as 20% of the people their birth weight is about 7 pounds. There's a very small number of people that are born with a much smaller birth weight of 2 pounds or even less, and a small number of people that are born at 10, even 11 pounds. Ouch. Sorry, and I've never even given birth, but I've seen it done, and I shudder to think what giving birth to an 11-pound baby is like. Okay, so most babies are born at about 7 pounds. A few are born much smaller, a few are born much bigger, but the mean is about 7. Okay, that's what that curve shows. What this curve shows is percent mortality, the percentage of babies that die. And I'm not sure where this data comes from. I want to say it's from the 1950s. So I think it predates a lot of the advances that we've made in keeping low birth weight babies alive. But there was a time when if you were born at about 2 pounds, right, there's 2 pounds, there's 2.5, there's 2.5 pound babies, your chance of mortality was about 70%. Today, I'm sure the figure's different because we're a lot better at this, but there was a time when most low birth weight babies simply didn't make it. Um, Babies that were born prematurely had very low odds of survival. Um, you also see that there with the high birth weight babies, babies that were born at 11 pounds, right there, had as much as a 15% chance of dying during birth, probably just because they're a lot more prone to damage during the birthing process because they're getting squeezed so much harder because they're bigger. Ow. Also, possibly they might be causing more damage to the mother, and that might put them at risk. So, low birth weight babies are likeliest to die, and high birth weight babies are likeliest to die. And babies with the lowest chance of mortality are the ones right here that are born at a little over 7 pounds. The mortality rate for those babies is only about 2%. Birth weight is partly heritable. People who are small babies tend to have small babies themselves, and people who were large babies tend to have large babies themselves. And so, since birth weight is partly heritable, this is a great example of stabilizing selection. Babies with extremely low or extremely high birth weights have a decreased chance of having more babies themselves that are like them. Babies that are in the middle are likeliest to grow up and make more babies that are also likely to fall in the middle. And so, this is stabilizing selection against the extremes, favoring the mean. And in fact, the mean birth weight is very close to the optimum birth weight, the birth weight that's at the lowest point on this curve. So stabilizing selection is selection against the extremes, favoring the mean. And then disruptive selection, great example of this came up in the 90s with work on, I believe this is an African bird called the black-bellied seed cracker. There's a black-bellied seed cracker. It's got a big bill and it, cracks seeds. And look at this lower graph right here. That's a graph of lower mandible length. Some of these birds have very short lower mandibles, and some have very long mandibles, and many of the birds are in between. Bates Smith looked at survival um, during, I think, a food shortage and found that the birds with short mandibles survived and the birds with very long mandibles survived. None of the birds in the middle survived. And the same thing was true for lower mandible width. The ones with narrow mandibles lived, and the ones with wide mandibles lived, but there weren't any birds in the middle. That's disruptive selection, selection that favors the extremes. Either the small or the large was favored, but the medium was not favored. My guess, I would have to go back and look this up, but my guess is there might be really small seeds and really large seeds in that bird's environment, but no medium seeds, meaning that if you were not specialized in either small or large, you, excuse me, you weren't going to be able to find any food. That's disruptive selection, favors the extremes of the range. Um, Favors the extremes of the range, does not favor the doesn't favor the middle of the range. And in the Galapagos, another one of the things that they've looked at, that the grants have looked at, is character displacement. On the very small island of Los Hermanos, one of the species present is Geospiza fuliginosa. And on Los Hermanos, it's not really a big enough population to show a very obvious bell curve, but that's where it is. And this is a measurement of beak depth. On the island of Daphne, there's a different bird species. It's not fuliginosa. I believe it's Geospiza fortis. Sorry, hang on. Let's go back. And on Daphne, fuliginosa and fortis, or fortis, has this bell curve distribution of beak size, and it overlaps with fuliginosa on Los Hermanos. On islands where the two don't coexist, they have very similar beak sizes. Over here on Floriana and San Cristobal, both of these birds coexist and the beak sizes don't overlap. What's happened is that where you have both species living together, selection favors fortis that has larger beaks. So it tends to push that bell curve in this direction. And in fuliginosa, selection favors birds with smaller beaks. So this bell curve gets pushed in that direction. And the reason is that this minimizes competition. If you are, let's say, fortis, the medium beaked ground finch, living on an island where there's also small beaked ground finch, then if your beak is large, it means you're not competing with the other species. So you're likelier to find enough food because there's less competition for it. So where these birds live together, selection has tended to push both of them to become less like each other. Where the two species don't live together, selection does not tend to make them less like each other, and so they overlap quite a bit. And we call that character displacement. And we actually get the same thing where we get large-beaked ground finches on the islands of Pinta and Marchena coexisting with medium-beaked and small-beaked ground finches. So that's character displacement for you. How we handle this is, let me see how much time we've got. I'm going to, okay, this is the last thing I'm going to cover. When we work with quantitative traits, what we measure in a population is a quantity called the variance. The variance in a quantitative trait goes like this. We take each data point, we calculate the average, we square the differences between each data point and the average, and we take the mean of those squared differences. That's the variance. If we take the square root of the variance, we get the standard deviation, which is a term I think you've seen before. Intuitively, you can think of variance as a measure of how wide the distribution is. If we have a population with a high variance, it means that bell curve is extremely wide, and many individuals might be very far away from the mean. If the variance is very low, that means the population doesn't vary much because all members of the population are very close to the mean, and that bell curve is very narrow. So you can intuitively think of the variance as the width of your bell curve. And we can represent that total variance by V sub P, P standing for phenotype. So if I went out and measured a bunch of, I don't know, birds in a population, and I measured, let's say, wingspan, I'd get a bunch of numbers and I could calculate the total phenotypic variance in all those numbers. Some of that variance comes from genetic variation in the population. Some of that variance comes from environmental variation in the population. And we can call these components V sub G and V sub E. We all vary in height. Okay, some of us are short, some of us are tall. So if I could measure everybody's height, I could calculate the variance. Some of that variance, some of what makes us have different heights is a result of genetic variation. You know, some of us have tall parents, some of us have short parents. And height tends to be inherited. Tall people tend to have tall kids. Short people tend to have short kids. Some of what makes us all different comes from variation in environment. Some of us probably ate very high quality diets with lots of protein in them, and maybe some of us were raised on Twinkies. Okay, that may be kind of an extreme example because I'm assuming most of you folks had fairly typical middle class diets. But that is a factor, that some of what makes people differ in height has to do with the food that they get while they're children. People that are starved as children often grow up to be shorter than they would have been had they not undergone starvation. So some of what makes a population of people differ in their height is due to variation in the environments that those people have been exposed to. And we can call those two components as V sub G and V sub E. And the total phenotypic variance you can think of as the sum of genetic variance and environmental variance. And you can kind of think of this as being like adding bell curves. V sub G would be the variance in a population if all of its members had an absolutely identical environment. If all of us had been raised in exactly the same setting and gotten exactly the same diet and the same exercise, then all of our variance would be due to our genes. There wouldn't be any other thing making us different. That would be V sub G. If all of us had identical genes, let's say we were clones, then the only thing that would make us different from each other would be variance due to environment. If we were all clones, but we were separated at birth and raised in very different circumstances, then the only source of variation would be variation due to environment, because we would all have identical genes. Sum those two, and what you get is total phenotypic variance, the sum of variance due to genes and variance due to environment. And we'll use that in a somewhat more sophisticated way when we reconvene on Friday. But I'm going to go ahead and stop the recording.